So we are going to the discussing about C3 plants and C4 plants. You can see the mechanism of the both. Already we are discussed about that. So how that pathways are takes place, C3 pathway and also C4 pathway. So now itself we are going to the discussion between differences between C3 plants and C4 plants. C3 plants and C4 plants. So these are the C3 plants. So next one C4 plants. So you can see the one by one. So the primary acceptor of the carbon dioxide in C3 plants is RUBP, that means ribulose 1,5 bisphosphate. But in C4 plants, the primary acceptor of the carbon dioxide is PAP, that means PAP, phosphoenol pyruvic acid. So what is the first formed compound within the C3 plants? That is 3-phosphoglyceric acid. 3-phosphoglyceric acid. So it having 3 carbons. That's why it is called C3 plants. In this one, the first formed compound is the oxaloacetic acid, that having 4 carbons. So the first formed compound are the primary acceptor of the CO2, ribulose 1,5 bisphosphate. It having ribulose 1,5 bisphosphate, that having the 5 carbons. But PAP, phosphoenol pyruvic acid, that having only 3 carbons. The first formed compound, 3-phosphoglyceric acid, having 3 carbons. Hence it is C3 plants. So first formed compound is the oxaloacetic acid, that contain 4 carbons. So that is the C4 plants. Next, in this one, only in C3 plants, only one type of cells are participate into the carbon fixation. Those are called mesophyll cells. In this one, the 2 types of So in this one, 2 types of cells are participate into the carbon fixation. One is called mesophyll cells and another one bundle sheath cells. So bundle sheath cells. So in this one, only one pathway is present, that is called C3 pathway. But here 2 pathways are present, C3 pathway and C4 pathway. C4 pathway takes place within the mesophyll cells and C3 pathway takes place within the bundle sheath cells. So C3 pathway takes place within the bundle sheath cell. But when we are observing C3 plants, that cranch anatomy is absent within the C3 plants. That cranch anatomy present. So that cranch anatomy present within the C4 plants. So these are generally present within the subtropical regions. So these are subtropical and also temperate regions. But these are highly active in tropical regions. So tropical regions. Next, you can see this one, cranch anatomy present, but only one type of chloroplasts. Only one type of chloroplasts are present. Those are granule chloroplasts. But it having the 2 types of chloroplasts. 2 types of chloroplasts, granule chloroplasts. Those granule chloroplasts are present within the mesophyll cells, and agranule chloroplasts are present within the bundle sheath cells. So food synthesis, that activity of the rubisco present within the mesophyll cells. So mesophyll cells lone rubisco activity ఉంటుంది. But ఇక్కడ చూస్తే మనకు that activity takes place within the bundle sheath cells, that rubisco present within the bundle sheath cells. Rubisco present within the bundle sheath cells. Rubisco activity takes place within the bundle sheath cells. So in this one, these are the major differences between C3 and C4 plants. Inka deenlo inka chusemanko. So what are the differences between C3 and C4 plants? The optimum temperature to the 25 to 30 degrees. So the optimum temperature to C4 plants, so 30 to 35 degrees. So, and also another most important waste phenomena. Next we are going to that discussion. That waste phenomena is called photorespiration. The type of photorespiration, that photorespiration is present within the C3 plants. That photorespiration is completely absent. Photorespiration is the completely absent within the C4 plants. Photorespiration is present in C3 plants. Photorespiration is absent in C4 plants. And also productivity. So these are highly efficient in high temperature, high light intensity, and also high temperature and high light intensity. The productivity is also, these are very less active in high temperature. So the productivity, when we are compared with the C3 plants and C4 plant, the productivity of the crop is more in C4 plants when we are compared with the C3 plants. So these are the major differences between C3 plants and C4 plants. The primary acceptor, RUBP, PEP. The first formed compound, 3-PCA. First formed compound is oxaloacetic acid. It having 3 carbons, it having 4 carbons. Hence it is C3 plant. This one is the C4 plants. So, and also these are highly efficient in high temperature. Photorespiration is present in C3. The type of photorespiration is absent within the C4 plants. So these are the major differences between the C3 plants and C4 plants. Example, most of the plants, dicot plants, are C3 plants. Datura, hibiscus, rice. So all are the dicot, all are the C3 plants. When we are observing that C4 plants, sugarcane, so maize, wheat. So all are the C4 plants. So those are major differences between the C3 plants and C4 plants. It is also one of the important 4 marks question in our IPE, and also most of the time that comparison is asking within the NEET examination and MSET examination. So NEET MSET lo ela vastayante, find out incorrect or correct regarding about that C3 plants and C4 plants. 2 types of cells are participate in C4 pathway. Only 1 type of cell is participate into the C3 pathway. So these are the major differences between C3 and C4 plants. Next we are going to another topic. In this one there are only 2 topics are there. One is called C2 cycle. That C2 cycle is also called photorespiration. The type of photorespiration is only present within the C3 plants. The type of photorespiration is completely absent within the C4 plants. So the optimum temperature to the C3 plants is 25 to 30 degrees only, but the optimum temperature to the C4 plants is 30 to 35 degrees. And those C4 plants are more active in high temperature. Okay, next we are going to the another topic, that is the photorespiration. photorespiration. It is also called C2 cycle. So this type of photorespiration, it is only present within the C3 plants. This type of photorespiration absent within the C4 plants. C4 plants లో photorespiration జరగదు. C3 plants లో మాత్రమే photorespiration జరుగుతుంది. So normally it is a one type of wasteful process. It is a one type of wasteful process, especially takes place within the C3 plants. It is absent in C4 plants. So normally you know very well, during that photosynthesis we are utilizing the CO2 and releasing the oxygen. We are in photosynthesis, so plants are utilizing CO2 and releasing the oxygen. But in respiration, plants or animals or any organism utilizing the oxygen and releasing the carbon dioxide. But in this one, in photorespiration, so in C3 plants, the plants are absorbing the CO2 and also lost their CO2. Ultimately, the productivity of the crop or plant, the rate of the photosynthesis is ultimately decreases. That's why it is called one type of wasteful process, especially in the C3 plants. So in the presence of high oxygen concentration. So name the cell organelle. Those are participate into the photorespiration. So in our textbook only 3 types of cell organelles are participate. So first one is chloroplast. Second one peroxisomes, and third one mitochondria. Third one is the mitochondria. But if maybe within the examination hall, it should be asking as a single option. Such a time, you can put the only peroxisomes. Name the cell according to your first year textbook. Name the cell organelle that is participate into the photorespiration. Only peroxisomes according to the first year 9th chapter cell is unit of life. Last topic microbodies lo peroxisomes lyoxisomes untayamma. Peroxisomes lo untundi. So it is mainly involved in photorespiration. So but it may be asking as a multiple option question. 3 types of cell organelles are participate into the photorespiration. So first one is the chloroplast, followed by peroxisome and followed by mitochondria. So what are the events those that takes place, especially in the C3 plants? When we are observing that first step in C3 plants, carboxylation, so the primary acceptor of the CO2 is RuBP. That RuBP is combined with carbon dioxide to form into two molecules of phosphoglyceric acid, each one containing 2 molecules of phosphoglyceric acid. The 2 molecules of the phosphoglyceric acid containing 3 carbons in the presence of enzyme rubisco. So you know the full form of rubisco? Ribulose biphosphate carboxylase oxygenase. So it is act as a both carboxylase and oxygenase. In normal photosynthesis in C3 plants, the carbon dioxide is combined with RuBP, ribulose 1,5-bisphosphate, to form into a 3-phosphoglyceric acid. Such a time that rubisco should be act as a carboxylase. But in high concentration of the oxygen, it may be oxygen high concentration undanko, that can be initiate the process of the photorespiration, ultimately decreasing the photosynthetic rate. That influence of the oxygen, actually oxygen gas is released during the process of photosynthesis. Photosynthesis lo manaki gas release avutundi, oxygen release avutundi, కానీ high concentration of oxygen, so that can be influence the activity of the rubisco. Such a time that rubisco should be act as a oxygenase. So in the presence of high concentration of oxygen, so that RuBP combined with carbon dioxide, So in the presence of high concentration of oxygen, that Rubisco should be act as oxygenase. It will producing into one molecule of the phosphoglyceric acid and another molecule of phosphoglycolate. So another molecule of the phosphoglycolate in the presence of oxygen. So recently 2020 NEET bit, how many phosphoglycolates are formed within the photorespiration when that oxygen is combined with ribulose 1,5 bisphosphate and adigarma. So one molecule of the PGA and another molecule of the phosphoglycolate is formed. In this one, that oxygen should be act as a oxygenase. Okay, so these reactions are takes place within the chloroplast. So then after that phosphoglycolate combined with oxygen to form into phosphoglyoxalic acid. So that is combined to form into phosphoglyoxalic acid. So that phosphoglyoxalic acid is enter into next cell organelle. That next cell organelle is the peroxisome. So in that peroxisome, that phosphoglyoxalic acid, in the peroxisome, glyoxalate in peroxisome, that is converting into another amino acid. That amino acid is called glycine. So that glycine having the 2 carbons, that's why it is called C2 cycle. That glycine is the amino acid. That glycine containing only 2 carbon atoms. So మనము ఫస్ట్ ఇయర్లో బయోమాలిక్యూల్స్లో గ్లైసిన్ ఫార్ములా చదువుతాం కదా. సో గ్లైసిన్ ఫార్ములా చూస్తే ఇలా ఉంటుంది, NH2COOH H. So only 2 carbons. So that is the structure of the glycine. So that's why 2 carbons glycine is present. So that's why it is called C2 cycle. So glycolate is formed. It is also called glycolate pathway. So that reaction takes place within the peroxisomes. That glycine is formed within the peroxisomes. Then after, so that glycine is entered into the another cell organelle. That cell organelle is called mitochondria. So that glycine is entered into the mitochondria. In that mitochondria, those 2 molecules of the glycine, combine to form into another amino acid. That another amino acid is called serine. So and also releasing the ammonia and also loss of carbon dioxide. Entry of the carbon dioxide into the plant body and also loss of the carbon dioxide into the plant body, out from to the plant body. That's why ఎప్పుడైతే carbon dioxide loss అయిపోతుందో, so ultimately the rate of photosynthesis is also decreases. The rate of photosynthesis is also decreased during the glycolate pathway or C2 pathway, are also called photorespiration. ఈ ఫోటో రెస్పిరేషన్లో ఇది తగ్గిపోతూ ఉంటుంది. ఇంకా ఏం చెబుతామంటే ఇట్ ఈస్ వన్ ఆఫ్ ద వేస్ట్ఫుల్ ప్రాసెస్ దట్ ఈస్ టేక్స్ ప్లేస్ విత్ ఇన్ ద C3 సైకిల్ అని చెబుతాం. ఎందుకు అంటే దెర్ ఈస్ నో సింథసిస్ ఆఫ్ గ్లూకోజ్. సో దెర్ ఈస్ నో ప్రొడక్షన్ ఆఫ్ ద ATP, దెర్ ఈస్ నో ప్రొడక్షన్ ఆఫ్ NADPH. అయితే ఇట్ ఈస్ ఆల్సో యూటిలైజ్ ద ATP ఫర్ దేర్ మెటబాలిక్ యాక్టివిటీస్. దట్స్ వై ఇట్ ఈస్ వన్ టైప్ ఆఫ్ wasteful process that is takes place only in the C3 plants. This type of wasteful process is absent in C4 plants. Okay, once again you can review this one. So in this process, in presence of high oxygen concentration, that rubisco should be act as a oxygenase and producing into one molecule of the phosphoglyceric acid, that is enter into the C3 cycle, no problem at all. But another molecule is phosphoglycolate. That phosphoglycolate is combined with oxygen to form into phosphoglyxalic acid. That glyoxylic acid is enter into the peroxisome. It is enter into peroxisome, that is converting into glycine. So that glycine or glycolate containing the two carbons, that's why it is called C2 cycle. Then after that, glycine molecules are enter into the mitochondria. In that mitochondria, two glycine molecules are combined to form into another amino acid. That amino acid is called serine. That amino acid is the serine, and finally releasing the carbon dioxide. And finally, lost their carbon dioxide. It is one of the wasteful process in C3 plants. It is one of the wasteful process in C3 plants because there is no production of glucose, there is no production of ATP, no production of NADPH, and finally it can utilize the ATP for their metabolic activity. Aite, this type of photorespiration is absent in C4 plants. How is it possible? Photorespiration is absent into the C4 plants because so there is no photorespiration is absent in C4 plants. How is it possible? So due to presence of two carbon fixation, C4 carbon fixation, C4 pathway, that is takes place within the mesophyll cells, and C3 pathway, that is takes place within the bundle sheath cells. So there is a special mechanism. There is a special mechanism to maintain the high concentration of CO2 that can be suppress the activity of the oxygen. Hence that rubisco should be act as a carboxylase. So that in the C4 pathway, that carbon dioxide is enter into the plant that can be released into bundle shape. So that CO2 is increasing the concentration of the CO2 level, intracellular concentration of the CO2 level, especially in mesophyll and bundle sheath cells, due to presence of how many? Due to presence of two carbon fixation. So in that first carbon fixation, that carbon dioxide is enter into the mesophyll cells, that can be increase the carbon dioxide level within the intracellular carbon dioxide level. So that can be minimize the that is minimize the oxygen level within the intracellular spaces of the mesophyll cells and also bundle sheath cells. That can be increase carbon dioxide level and minimize the O2 level such a that rubisco is act as carboxylase. That rubisco act as a carboxylase. So that can be suppressed, that can be decreasing the level of oxygen between the mesophyll cells or within the mesophyll cells due to presence of two carbon fixations. That's why that rubisco should be act as a carboxylase but not oxygenase. It may be that should be act as a oxygenase that can be initiate the photorespiration, but due to two carbon fixation that can be increase the carbon dioxide level between intracellular space and intracellular carbon dioxide level is increases, that can be minimize the level of oxygen. Hence rubisco should be act as a carboxylase only. So that's why there is no initiation of the photorespiration, especially in C4 plants. So that's why C4 plants are high efficient in carbon fixation or photosynthetic rate when it is compared with the C3 plants. So that is the photorespiration. Recent 2020 NEET bit vachindi. So when that oxygen is combined with C3 RBFBP, how many molecules of phosphoglyceric acid another, one molecule of the phosphoglyceric acid and another molecule of the phosphoglycolic acid under the influence of oxygen. So this influence of the oxygen on C3 plants was first discovered by me book lo ledu, one of that scientist is named Warburg. That effect is called Warburg effect. That effect is said to be Warburg effect. Okay, so this is the another topic, that is photo respiration, also called C2 cycle. It is also called glycolate pathway. So it is only present in C3 plants, it absent in C4 plants. Next we are going to the another topic. That topic is called factors affecting the rate of photosynthesis. The factors which are affecting the rate of photosynthesis. So the factors affects the rate of photosynthesis. What is the rate of photosynthesis? Means the end product. What is that end product? Is glucose. Glucose converting into sucrose. Sucrose is converting into starch. That starch should be act as a energy source in all plants. So a starch lone manaki energy source untundi. So avanni form avadani the rate of photosynthesis annam. So that rate of photosynthesis is influenced by to the many factors. So those are broadly divided into two types of factors. Internal factors are also external factors. So internal factors are also called plant factors. Internal factors are also called plant factors. So what are the plant factors? Very simple. Chlorophyll content. So amount of the chlorophyll, number of chlorophyll molecules, amount of the chlorophyll, number of chlorophyll molecules, and also number of chloroplasts in each mesophyll cell, and also internal CO2, the concentration of the internal CO2, and also number, size, shape, structure, or everything about the leaves. So number of leaves, size of the leaves, and also amount of the chlorophyll within the leaves, orientation of leaf, that means arrangement of the leaf, arrangement of the chloroplast within the mesophyll cells in previously. So when those chloroplasts are arranged within the mesophyll cells parallel to the along with the cell surface, that can be get more amount of the incident light. Maybe those are arranged in perpendicular to the cell surface, that can be get the less amount of the sunlight. That's why, so that in the size, shape, amount of chlorophyll, number of chloroplasts, arrangement of chloroplasts, alignment of the chloroplasts within the mesophyll cells, internal CO2 concentration, all are considered as internal factors. Those internal factors does not explained individually in our textbook and also in +1 book. So next we are going to the external factors. The external factors which are influence the rate of photosynthesis. First one, very well, light. Second one, temperature. Third one, CO2 concentration of the CO2, and finally that is the water. So these four are the more important one to discussing about the external factors. Those are influence the rate of photosynthesis. So first one, light. So the light can influence the rate of photosynthesis in three ways. So one is quality of light, quality of light, another one intensity of light, and third one duration of light, duration of light. So before going to the external factors, one of the important 2 marks question is there, that is called law of limiting factor, law of limiting factor. So that law of limiting factor was proposed by F.F. Blackman. Law of limiting factor was proposed by F.F. Blackman. If the any system or maybe photosynthesis is influenced by to the many factors like light, temperature, concentration of oxygen, water, so the low concentrated factor is highly influence the rate of that particular process or rate of photosynthesis. That type of factor is called limiting factor. That low concentrated factor, that minimal value factor, that factor is called limiting factor. That is called law of limiting factor, was proposed by to the everyone day F.F. Blackman. Then we will come to the external factors like light. So first one intensity of light and the quality of light and the duration of the light. So first of all you can see the quality of light. So the photosynthesis takes place only in the visible light. The photosynthesis takes place only in visible light. Okay. So quality of light. Normally photosynthesis takes place within the visible light. That is simply called PAR, photosynthetically active radiation. So photosynthetically active radiation, it takes place only in visible light. That visible light ranges from 400 to 700 nanometers. Actually 360 nanometers to the 740 nanometers. But for our convenient purpose, 400 to 700 nanometers, that is visible light, that is highly suitable to the photosynthesis. In this one, you know very well that VIBGR. So in that VIBGR, the most suitable lights to the photosynthesis is red light and also followed by blue light. So in another lights, there is a small amount of photosynthesis, small rate of photosynthesis, but there is no negligible photosynthesis in green light. Negligible photosynthesis within the green light because those chlorophyll molecules can reflect the green light. So that is the quality of the light. Most important suitable lights to the rate of photosynthesis are red light followed by blue light. When compared with the red light and blue light, the rate of photosynthesis is also more within the red light when compared with the blue light. Okay, so that is the quality of light. Next, intensity of the light. If maybe intensity of the light is increases, the rate of photosynthesis is also increases in certain levels. If the light intensity is increases, the rate of photosynthesis is also increases. That means directly proportion. But remembering once again, but in high light intensity, those chlorophyll molecules are undergoes to photooxidation. Those chlorophyll molecules are undergoes to photooxidation, are also called photobleaching. So what is the meaning of photooxidation and photobleaching? In high light intensity, those chlorophyll molecules are undergoes to splits up, breakdown of the chlorophyll molecules in high light intensity. Those are called photooxidation or photobleaching. But how that chlorophyll molecules are protecting from this dangerous phenomena? With the help of accessory pigments. You know that one chlorophyll A is the main important photosynthetic pigments. Chlorophyll B, xanthophyls, carotenoids, and also phycobilins all are present along with the chlorophyll. So these are involved in dual role. One is absorbing the light energy that can supply to the reaction center, and also that necessary pigments can protect the chlorophyll from photo oxidation or photo bleaching. Then after duration of the light. So how much duration of the light is required to proper activation of the photosynthetic light reaction? So the duration is not required. So low light intensity, so continuous is not sufficient to the photosynthesis. Only intermittent light, flashlight or intermittent light, that is highly sufficient to the rate of increasing the rate of photosynthesis. That's why when we are compared with morning time and evening time, so on that midday time, the rate of photosynthesis is very high because plants can get more amount of the sunlight. Because that light is essential only to the activation of the electrons, only to the activation of the chlorophyll A molecules, those are present within the reaction center. Reaction center lo unna chlorophyll molecules activate avadam kosame light energy kavali. So when only that nanosecond light is enough, nanosecond light duration, so light duration is enough to the activation of the chlorophyll A molecule. When it is when that chlorophyll A is undergoes to activation, so immediately that is in exciting state, immediately lost their electrons. Those electrons are entered into the different electron acceptors already that was discussed within the cyclic and non-cyclic electron transport. So that is the another important thing. Next, come to the temperature. So you know very well, lot of enzymes are participate into the C3 cycle, C4 cycle, and also last step in the non-cyclic electron transport. All enzymes are active, so within the increasing in temperature. When the temperature is increases, the rate of photosynthesis is also increase up to 40 degrees or 45 degrees. After onwards, those enzymes which are participate into the photosynthesis, all those ones are undergoes to the denaturation. That means enzymes are made up of proteins. Those proteins are undergoes to the denaturation. Mari, how much that optimum temperature to rate of photosynthesis? So in C3 plants, the rate of photosynthesis is optimum temperature to the C3 plants is 25 to 30 degrees. But the optimum temperature to the C4 plants 30 to 35 degrees. So which ones are more efficient in high temperature? C4 plants are more efficient in photosynthetic rate in high temperature when compared with the C3 plants. Okay, that is and also in low temperature, some of the plants like that was not there in your textbook. So in low temperature, so some of the conifers means gymnospermic plants which are found on that high mountain regions. Those are also able to photosynthesis in -6 degrees. In -6 degrees. But some of the sulfur bacteria, so the sulfur bacteria are present in hot springs more than 70 degrees. Those are also able to photosynthesis. This is the effect of the temperature on photosynthesis. And next come to the most important one, that is concentration of the carbon dioxide. So you know very well that carbon dioxide is the main important raw material. Important one, concentration of the carbon dioxide. The concentration of the carbon dioxide. So the main important raw material to the process of the photosynthesis is the carbon dioxide. So that carbon dioxide is mainly helpful to the synthesis of the food material. So the type of carbon dioxide, if the carbon dioxide concentration in atmosphere is 0. The carbon dioxide concentration is atmosphere is 0.03 to 0.04. If maybe that carbon dioxide concentration is increased up to 0.05 or 0.5, the photosynthetic rate is also increases. The photosynthetic rate is also increases if the increasing the carbon dioxide concentration. So carbon dioxide concentration is increases up to 0.5, that photosynthetic rate is increases. But in high CO2 concentration, there are two important steps that can be decreasing the rate of photosynthesis. First one, that high concentration of the CO2 can favor the closing of stomata. So in high concentration of the CO2, those can be favor the closing of stomata that can obstruct the gases exchange. That is also one of the important thing. And another one is, so that high concentration of the CO2, so that should be act as a poison to the cells. That should be act as a poison to the cell. That is also decreasing the rate of photosynthesis. But that CO2 concentration, that activity of the influence of the CO2 on the photosynthesis, it is varies in C3 and C4 plants. You know that one CO2 compensation point. So CO2 compensation point means the concentration of the CO2 is released during the respiration, and to the concentration of the CO2 is utilized in photosynthesis. So in which point the rate of the concentration of the CO2 is equal to the concentration of the CO2. Those are released in both. So released in respiration and utilized in carbon dioxide. Means the amount of the CO2 released during the respiration is equal to the amount of the CO2 utilized in photosynthesis. That is called CO2 compensation point. So that CO2 compensation point, the CO2 compensation point, when you can see this one, that CO2 compensation point is more to the C3 plants and less to the C4 plants. Enduku ante C4 plants are utilized more amount of CO2 for their photosynthesis because due to presence of two carbon fixations. Due to the presence of two carbon fixations, they can they can utilize more amount of CO2 when compared with the C3 plants. That's why the CO2 compensation point is very, very less in C4 plants when compared with the C3 plants. So that's why, so the C3 plants are more active in high CO2 concentration. So C3 plants active avvali ante ekku CO2 level kavali. Andike that's why, so those C3 plants like bell pepper and tomato, those crops are greenhouse crops and those greenhouse crops can be grow within that area containing high CO2 to proper activation of the photosynthesis because that CO2 compensation point is very high to the C3 plants. And the last one, come to the water. So that water is highly influenced the rate of photosynthesis. That water is highly influenced the rate of photosynthesis. It may be water availability is present, that is involved in more processes within the photosynthesis. Water donating the electrons, protons, oxygen. The proton donor, electron donor and oxygen donor is the water. And also that if maybe that water is present within the photosynthetic mesophyll cells, that is involved in many metabolic activities, cell growth, cell division, cell expansion. Every metabolic activity takes place within the water molecule. If the plant facing water stress conditions, If the plant facing water stress condition, ultimately the photosynthetic rate is decreases because the cell is undergoes to the affecting the cell growth and cell metabolic activities. And also it may be cell facing the water stress condition, immediately one of the phytohormone, one of the plant growth regulator, that is called ABA, abscisic acid. It is the natural anti-transparent. That concentration of the natural anti-transparent is increases in the leaves, that can favor the closing of stomata and obstruct the gases exchange. And finally, photosynthetic rate is decreases. These are the major and main important things. Those are occurs during the photosynthesis. It is very, very essential to know about the photosynthesis. Okay, thank you very much.